announcement. The Reinsch Lectureship, for those of you who may or may not remember or are new, is in honor of the first UC Davis graduate who decided to go into surgery, and tragically, a few months before finishing his general surgery residency, was in a car accident on his way to answering a patient call. So this lectureship was established in his name. And it's appropriate to think about this at a time when we are celebrating the selection of our new residents and the important role that the house staff and the residency plays not only in teaching one another, but in the care for our patients. And just as I was so impressed with M&M this morning and the issues around the ability to rescue, and I think that that's what an academic medical center and a place that has house staff around all the time really brings in contradistinction to many of our private hospitals. So the role of residents cannot be underestimated and not appreciated enough. So today's lectureship is really in honor of all of you and a reminder to all of us to take a moment to thank you. With that, I want to bring up Dr. F.A.C.S, who, as I announced earlier to some of you who weren't here, recently received accreditation for her Rectal Cancer Center of Excellence, and the whole colorectal team really deserves great credit for that. And we will, another round of applause for that. It really is a big thing. And she was responsible for sponsoring this year's Reinsch Lectureship, so I'm going to turn the introduction over to you. Thanks, Dr. F.A.C.S.
Who I really want to thank, who's not here, is our nurse navigator. I think a lot of you know Emily, and she really poured her heart into helping us with our accreditation. And so when you guys see her, please thank her. She's the true heart of that program. So I'm very happy to sponsor Dr. Alverdy. As you know, colorectal surgery is one of the youngest divisions, so we have no named lectureship. So when the call came out for this, I grabbed it right away, because right away I can think of no one else I would want to give this lecture but Dr. John Alverdy. I have known him since 1988. Of course, I was in grade school. He was in junior high at that time when we met. But actually, he interviewed me for surgical residency. And he has just been phenomenal in watching his career grow. He is a Loyola grad, and I know there's a few of us that are Loyola grads here, either of medical school or of residency. And then he did his residency through Michael Reese, which was affiliated at University of Chicago. And he's now vice chair of surgery at University of Chicago. He is director of the surgical infection and therapeutic program. And the reason that I chose him is not only because he's a great academician, he's a great speaker, he's just a good guy, but also this is a talk that no matter if we're in surgical oncology, acute care surgery, colorectal surgery, if you're dealing with cancer in the intestines, this is a wonderful talk that is going to go across all of our subspecialties. So I'm very pleased to introduce Dr. John Alverdy.
Thank you, Dr. F.A.C.S. It's a great honor to be here and speak with you. I want to thank Dr. Farmer, Dr. F.A.C.S., Dr. Jerkovich. Your leadership is outstanding. You should know you're being trained in one of the best programs in the country under outstanding leadership. And again, I thank you for the honor. These are the people that have done all the work and the funding. What I'm about to show you is going to seem like a lot of work in terms of bench molecular science that's translated into clinical science, and it can't be done without really young, smart people. Now, I'm not that smart, but I'm surrounded by really smart people, and that's the key to a successful career as an academic surgeon, is that you need to be able to create chemistry, excitement, enthusiasm around you, and then create a team of people that are excited to work with you. You can't do research by saying, okay, I have a great idea. Let's get pancreatic cyst fluid and drop it off in the proteomics lab and say, hey, take all this stuff and make a graph so I can give a poster in Paris. That doesn't work. You've got to really be engaged with your collaborators and be able to attract great people, like I was able to attract Katie Romanowski to my lab, who spent 2 years there. Hi, Katie. So what does a gastrointestinal surgeon think about right before they go to sleep? We think about a lot of things, like I hope my patient will not leak. I hope my patient will not develop an ileus or an obstruction. I hope my patient will not develop a serious infection. These actually are the 3 most common, costly, and deadly complications that face us today. And every surgeon has ghosts. Dr. Farmer's shaking her head there, because we all sleep with these ghosts, these cases that we wish we had just done something. And this idea of failure to rescue, which actually came from the military, is really, it really embodies what you all do great. At 3 o'clock in the morning, a second-year resident will see a patient and be nervous and worried and anxious, and that is what the difference is between this place and a private hospital. And so I call Surgical Morbidity and Mortality Conference, it's a place where we learn from the ignorance of experts. And I don't use ignorance pejoratively. Ignorance means we don't have facts like that PE case, the Caprini score we're talking about this morning. Things are highly probabilistic. They're not deterministic. We got a score, and you got a drop-down menu, and then you put a bunch of things in there, and you raise a score, and it's probabilistically predictive, but not inclusive of all cases. And the problem with the PE is people die if they get it. So the risks are very high. The stakes are high. But unlike the aviation industry, we rarely arrive at the root cause of a complication. We just had a bad airplane crash, right? And they ground the planes. And they're getting the black box, and they're looking at satellite images. We don't do that. We get a 5-minute discussion, we all leave, and we know what that anastomotic leak was. Tension, ischemia. Because Dr. So-and-so said it. Okay, tension, ischemia, whatever you say. But there are things that we just don't understand. So microbiome sciences is nothing new. Ecologists have known this for 50 to 100 years. That bacteria live in complex communities. The difference here is that we can now measure it. We can't culture 80% of the stuff on us, but we can now sequence it, take the genes and the contigs and run them against a database and actually know who's there. And what's so fascinating about it is that it's a dynamic microbial consortia. It's not one bug. Everything that goes in your mouth and your patient's mouth is metabolized by your microbiome. And those exoproducts, those synthesized products, those metabolites, some of which are very small, have profound effects on your body at the level of the epithelium. You don't have to translocate the bacteria. You don't have to become bacteremic. Right here at the top, those metabolites can seep through your epithelium, and they can affect what are called HDACs, histone deacylation events, on the genes and change not gene structure but gene transcription at the epithelial level, which then has not only local effects on things like macrophages and dendritic cells and cytokines, but systemic effects. So what you eat changes your microbiome, changes your immune program. Your immune system is sort of a puppet, if you will, of your microbiome. So we can't understand the root cause of surgical infections, for example. 50% of all cases at M&M, I kind of looked recently, are really related to an infection of some sort. Yet infections like, somebody dropped something in there, you didn't prep right, you gave the wrong antibiotics, not that simple. But the whole is greater than the sum of the parts. So now we can take a complex microbial community from urine, CSF, poop, a wound, and we can sequence it and get the community structure and membership, but that's not enough. We need to know not just who's there and under what structure they're there, but we need to know what they're doing. What metabolites are they making? We can do metabolomics. What proteins are they making? How are their genes being transcribed, turned on and off? But it's not enough because the whole is greater than the sum of the parts. So now you have to take that whole community and you have to put it in germ-free mice to phenocopy the actual problem that you're trying to understand. You colonize these mice, for example, with the actual human microbial community. So patients with an ileus, adhesions, or leak. Imagine a world in which we understand why people get an ileus and adhesions and leak. Like 80% of our complications would be gone. But that's not what we study in general. So look at this study from PNAS published last year. They take the gut microbes from 34 monozygotic twins, discordant for MS. One twin has it, the other doesn't. They take their poop, they put it into germ-free mice, and only the mice that got the poop from the affected twin developed encephalomyelitis in the mice. Whoa, how did that happen? Oh, which bacteria was it? Which one seeped out? Did they do a blood test? Did they find a virus in the brain? Nothing. Think of the dimensionality of that poop on the epithelium, on the in cells, making metabolites, changing gene expression, reprogramming them. It's too hard to track. N dimensions of understanding. How about ileus? Oh yeah, we know it is. This is a very good group from Leeds. I know Dr. Jane. I met him recently. And this is a British Journal of Surgery. It's a very important European journal, highest impact. Here's the review article. They never mentioned the word bacteria in the entire thing. Oh, ileus, yeah, that's from opioids. You don't beat up the bowel too much. You know, be careful in their economy of motion. Yet, when you look at the evidence, this excellent study from Gut is their highest impact GI journal. This group has done several studies, and what they've shown is that postoperative ileus starts right up there. Bacteria aggregate on the epithelial surface. They can then communicate to macrophages and dendritic cells, and those cells land right on synaptic junctions and actually cause paralysis. And they showed it in germ-free mice, knockout constructs, etc. And so, you know, again, is the immune system a puppet of the microbiome? Now, we do a lot of Whipples at the University of Chicago, and I'm really getting sick of hearing about the delayed gastric emptying after a Whipple. You know, I hook it this way, that way, I do all that. Table, post-pyloric, pre-pyloric, no, the vagus, this. I mean, what do we know? Ignorance of experts. They haven't solved the problem. Everybody gets an ileus. I don't know. You know, okay? What about adhesions? You know, imagine a world in which you discover adhesions and you know why they occur and you've got the drug for it. You would win the Nobel Prize. But that's not what we study. But adhesions, well, I tell you, that's due to, you know, microvascular, inflammatory cells. You beat up the bowel, collagen, VEGF, TNF, oh yeah, that's all bad. Except this stuff focuses on serotonin. And this showed in a knockout mouse that if you don't make serotonin in the GI neural plexus, you know, you don't get the ileus. Turns out that the most important secretagogue for intestinal serotonin are the GI tract bacteria. And then that same group from the British Journal of Surgery forgot to read this paper from 1999 from a Swedish group that showed if you take a rat and you beat up its bowel, it gets adhesions. But if you raise it germ-free and beat it up, there are no adhesions there. It's kind of like fetal surgery, right? Where's the scar? What happened? But no bugs, no adhesions. Then they put the bugs back into those mice, and adhesions form after you rough it up. So when you think about this, ileus, adhesions, I'm going to talk to you a little bit about leak. Bacteria are the root cause. But as James Shapiro, who is a collaborator of mine at U of C, said, there really are no units, there's just interactive systems. But see, in medicine, especially with biologics like Remicade and these other drugs, we like the agonist, the receptor, and the drug. And nothing in life is that simple, or in biology. So an anastomotic leak, it's the Achilles heel of the GI, of surgeon. 2 people suffer when a leak occurs, the patient and the surgeon, and I have never met anyone that hasn't had a leak, except for me. I've never had a leak. But, leaks are bad because they cause infections, and infections actually change, we think, the oncologic outcome for patients. Patients that have an infection, an intra-abdominal infection, don't do as well. But we're sure we know who leaks because, we know things other people don't know, as Trump would say. I know things other people don't know. So this study is a great study because it's from the Netherlands, from Holland. And they looked at cases where they said to the surgeon, okay, you're doing a colon resection. You know your patient, you know their albumin, how many units of blood, how long it took. Look down and give me, on a scale of 0 to 100%, you tell me what you think the probability of a leak is. They can't predict. The ones they thought were going to leak didn't leak. So at M&M, we go, well, this guy was fat and he was old and, the case took a long time and, they've forgotten confirmation bias of all those same patients that didn't leak. But in this particular case, especially in the low incidence cases like PEs, wound infections, and that's we're sure we know why that patient leaked. We don't do like the aviation industry, a root cause analysis. We just kind of get it out here. So surgeons, not always right, but never in doubt. Okay, so here are 3 unfalsifiable assertions that make most surgeons angry. In expert hands and high-quality institutions, there's no evidence that inadequate technique is the major cause of an anastomotic leak. I'm happy to entertain any argument, have public discourse on this. I've never lost the argument. Nobody can provide any evidence for that. Of course, bad technique will cause a leak. But the problem is there's no evidence that inadequate technique explains every leak, or even the majority of them. So you say, well, that's patient-related factors. I'm a good surgeon. I train at the best places. That stitch in there. So you say, well, this patient was fat, got neoadjuvant chemo-RT, weight loss, frail, psoas muscle was thin. The problem is, probabilistically, most of that group of patients you just described don't leak. But the one that leaks, that's from the obesity. The one that didn't leak, I'm a good surgeon. I know what I'm doing. So confirmation bias hasn't done it. So my work really tries to understand at the most fundamental molecular level what the root causes of our complications are. And this requires sort of bench-level biology mixed in with some clinical context of that biology that you're studying and some translation ability of that in patients. So this is Ben Shogan. He worked with me for 2 years in the lab when he was a resident, then went up and worked with Heidi Nelson to do a colorectal fellowship, and now he's back in my lab. And he did this very nice work looking at the inflammatory signals that activate F.A.C.S, one of those bugs that you can't kill. The only way to get rid of F.A.C. in the gut is to boil the gut in water for 45 minutes and autoclave it, and you can't do that. And bacteria actually sense these signals, and they activate their virulence genes, and then they adhere to tissues that are injured. And what's really interesting is Ben showed that this F.A.C. strain that we call collagenylytic, it can break down collagen. It does it 2 ways: directly by secreting an enzyme, collagenase, that can break it down, and indirectly by shifting host tissue proteases or collagenases that take the MMP9 from its inactive form to its active form and break down the collagen. And so these microbes, they can express what we call emergent phenotypes. There's the observer effect, like the Heisenberg uncertainty principle. You take the bug out of its environment, grow in ideal growth conditions, and you lose the very phenotype that you're trying to understand. So you have to understand these things in the very environment, because environment regulates genes, and genes get transcribed. And yes, everything we do is important. Look at that. If you do a fast, bloodless, planned, well-done operation, you're going to release less of these environmental cues and turn on less of these bacteria. And so, yeah, it's important. Technique's important. But across all great institutions like UC Davis, people still develop leaks, and we don't understand why. Now let's look at our mouse models, because a lot of times I'll say, well, I operate on patients, not mice. And what you're showing us doesn't really translate, but it does. Let's look at this. This is mice that either got placebo or we did things to them, gave them morphine, devascularized the anastomosis, exposed them to a lot of antibiotics. And here's what we learned. 100% of the mice get colonized on the anastomotic tissues with E. faecalis. Well, how many mice actually leak? Because it's really hard to make a mouse leak. You can just do a bad job and they will not leak clinically or even microscopically. But among those that do leak, they're only colonized by collagenolithic E. faecalis. So we did all the homework on this. We knocked the gene for collagenase out of the E. faecalis, no leak. We put collagenolithic and non-collagen back into germ-free mice. No, it all worked out exactly the way it should. But most of the leaks that we saw in these mice, this is what's really interesting, are subclinical. The omentum's wrapped around it. You see this little dilated bowel here? When you pull that bowel off, there's a hole. But that mouse is perfectly fine. Like your patient comes back with an ileus, you're like, oh boy, put an NG tube down, give some antibiotics, sit tight, it goes away. Was that a leak? No, it wasn't a leak. Okay. No? I swear, there's no way that leaked. Okay? Only 10% of the mice in our study, these are our models, it's really nice, actually leak. Now look at that abscess there. That's 10%, okay, have an abscess or peritonitis. That's really low. That's about what you see if you do sort of high-risk LARs. It's about 10%. But remember that patients only come in and tell you they have a leak when they are sick. You don't know about 80%, the unknown unknowns. So if we only count the leaks that manifest symptoms, we cannot arrive at a root cause of the problem. So this is the latest and greatest from the lab. This is work by Rick Jacobson, who's spent 2 years in my lab. He's a surgery resident at Rush and saw me give a talk and wanted to work in my lab for 2 years, a brilliant researcher. And he said he studied it when he was at Vanderbilt. He studied plasminogen system with an orthopedic surgeon and said, I think plasminogen plays a role in this leak, and I want to use your model to prove it. Now, tissue protease activity, and there are many proteases in tissues, is the hallmark of healing across all wounds. Remember, you make collagen, you lay it down, it makes things strong, but then you got to remodel that collagen. So you synthesize, degrade, synthesize, degrade, synthesize, get those cross links nice and hard, and then the thing heals perfectly. Too much protease, things are going to break down. Not enough, things aren't going to be strong. So the body gets it just right. And one protease is plasmin, because you have clot and fibrin, and you need to break that down the same way you break down collagen. And so it turns out that Rick showed very nicely that macrophages make a exoproduct, a soluble product that can shift the proform of UPA, which is the urokinase, to its UPA, and shift plasminogen to plasmin in the wound. And he showed very nicely any wound you make in the body, if you can stain it like he did, you'll see plasmin and plasminogen. Plasminogen, this is a plasminogen assay. He radio-labeled it, fluorescein labeled it, and injected it systemically, then made wounds in the gut. And you could see it aggregates right away to the anastomotic wound. So then he started studying E. faecalis, because we have strains that make collagenase and strains that don't. We have all the different strains, and they're bioluminescent. And what he was able to show is that local environmental cues like opioids, pH, exposed collagen turn on the E. faecalis. It expresses more collagenase, and it actually starts activating this system. And he's done so much molecular detail. This is in Cell and Review. And he showed that not only does this take the pro-MMP9 to the MMP9, but MMP9 itself activates plasminogen, forcing it to plasmin, as does E. faecalis at that enolase step there. So here's the really great part of this story, because this is what everyone likes. There's a drug that will block enolase activation of plasminogen, and you use it all the time in trauma. It's called TXA, tranexamic acid. So if this hypothesis is correct, the TXA should completely block this process, and it does. it basically dampens that iterative loop that activates all this. So then he put it in our mouse model, and it's 100% protective against an anastomotic leak, no matter what pathogen you stick in there, F.A.C.S, Serratia, Pseudomonas. And this is topically applied TXA. So we're now working on designing clinical trials where we take a adhesive form. It's in like a gel form where we put this on there, and you can prevent an anastomotic leak. How do we do this? Bottoms up. Study the problem at the biologic level. Try to understand what's going on, and then apply. Let the basic science inform the therapy. Okay. So again, I want to talk to this issue of what is probabilistic and what is deterministic. Because at M&M, we always talk about the probability of this occurring. Well, who should get Lovenox? I'll tell you what I learned this morning. I think you kind of got to give it to everybody. It's like prophylactic antibiotics. Who should get them? Well, I don't know. Give them to everybody. Now, we know that for a lap chole, you don't need prophylactic antibiotics. For an elective lap chole without inflammation, prophylactic antibiotics are unnecessary. We give them at U of C. Everybody gives them. Everybody gets them because it's pretty benign. 1 dose of cefoxitin or something, cephalo du jour, whatever you guys use here. Okay, so here we go. Here are all the things that probabilistically lead to a leak. Oh yeah, smoking, radiation, NSAIDs, blood loss, ischemia, yeah. But it all affects the microbiome. But these steps need to happen, and we need to dissect them out. You have to have in vivo virulence activation, which Katie studied when she was in the lab with me, adherence to tissues, immune escape, activation of these proteases. A lot has to happen, and that's why the probability of leak is a low probability event. It's a low probability event. And the key, and remember, we only count the leaks that become clinically manifested. So what's important here? Don't disturb your microbiome, because if your microbiome can recover quickly, I'll show you some data in a minute, well, it will competitively exclude things like F.A.C.S and others. And also, the local cues that activate these microbes need to be kept at a minimum. So we have to preserve our microbiome and really be good surgeons. And everyone here, your leadership tells you that every day you operate with them. Did you prep this patient properly? Did you come to clinic and see that patient? Did you make sure that that patient got proper biopsy? Did you give that patient preoperative chemo RT? Have we done everything we can to make this patient in the perfect place? Yes. Then you do this beautiful operation with Dr. Farkas or Dr. Farmer where there's economy of motion, gentle handling of tissues, and there's experience and a rhythm and a cadence to that operation because of their experience and their dedication to their craft. And all that matters down here. But we can't ignore this other piece because we see our best surgeons have complications, that there's something else that we haven't accounted for, and it's biology. And biology is fundamentally uncertain. So the more we can know, the more we can do that. So this is Bader Shakshir. Dr. Romanowski probably remembers Bader. Bader is now at WashU doing a pediatric surgery residence, spent 2 years in my lab, and he showed that morphine in this model will disturb the microbiome very profoundly. And that when you take strains of F.A.C.S and you just dump morphine on them, or fentanyl, virulence genes are turned on. That's interesting. So here comes ERAS, an empirical shot at telling us what to do right and wrong, which is okay. But we still need to understand why all these enhanced recovery programs are good for the patient at the most fundamental biologic level that we can. Because basically its edict is give food and avoid opioids. Feed early, don't use opioids, right? And we know this whole opioid crisis is a problem. And we also know that our patients that are on prolonged opioids probably don't do as well. So I couldn't be more delighted to speak to you and thank Dr. Farmer, Dr. Farkas, Dr. Jerkovich for the honor of this podium and its privilege, and I'd be happy to answer any questions. Thank you very much.
Well, first of all, before we open to questions, I just want to thank you again for coming.
Wow.
It takes a minute to appreciate this, but this is our little, if you noticed when you came in,
Yes.
our DNA that's sort of
Yes.
hanging in our entryway here is on the back of your, this is
Oh, yeah.
a weapon, but we do know that you can get through the security system to
Okay.
get this back in the airport.
All right.
But just really want to thank you so much for being
much.
here.
Thank you, Dr. Farmer.
We'll stop to take pictures a little bit at the end, although I love it that he sneaks up there. He's so good.
Yeah. The paparazzi follow me around all the time.
So this is absolutely spectacular presentation, and it really does bring home, and you do it in a just masterful way, make complex science really understandable to us. You know, what do they call us? The non-cognitive specialty surgeons. But how much science influences everything we do. So I think I'm going to take the moment to end this fantastic conversation. We are going to have some time for Dr. Alverdy to spend with the residents. And I just want to thank you again for a spectacular time.
Thank you.